Okay, I think we can get started. I'd like to welcome everyone who's viewing us on our webinar from the Poland Silicon Valley Center. And I'd like to welcome Dr. Priebe, who is from the MD Anderson Cancer Center in Texas. I am in sunny California, and it's 7 a.m. on Wednesday morning here, 9 a.m. in Texas, and 4 p.m. in Poland in the afternoon. So I have a feeling it's dark and cold where most of you are. We're happy to have you here on our webinar, and I'm very excited to be able to have this discussion with Dr. Priebe, who has had a remarkable career in drug discovery at MD Anderson. So I'd like to give just a small background about Dr. Priebe. He started out at the University of Warsaw, or that's how we pronounce it here, or Warszawa. And since that's my namesake, I'm always happy when someone starts out at that university. So welcome, Dr. Priebe. Dr. Priebe went from University of Warsaw to Ohio in the United States, and then to MD Anderson Cancer Center. He is also a professor of chemistry in Poland, even today. He's been focused on design and development of drugs, selectively targeting DNA for signaling and metabolic pathways aimed at cancer progression. He's had several drugs that are in clinical trial and others that are in preclinical development. He stresses collaboration between scientists in academia and industry, which we will talk about. And he definitely will be an incredible role model for all of you attending and others in the scientific academic community. So, Dr. Priebe, welcome again.
Thank you for the introduction.
What made you decide to stay in the United States for your career instead of going back to Poland after you did your postdoc in Ohio?
Really, there was not like a single moment to make such decision because, just as you might remember, that was a state of war at that time, just in Poland when I was a postdoctoral fellow. So really, we were waiting for some changes and freedom coming back to Poland, and it was prolonged periods of time, looks like. So just you had to do something with yourself. So we decided to prolong stay, but I decided to stay only when I can get an independent position here, and I was lucky to get a tenure track position at the University of Texas MD Anderson Cancer Center.
Are you happy with that decision, or do you think you could have done what you have done in your career in Poland?
You never know what will happen. There's different routes in a career, but definitely here, landing in the #1 cancer center in the United States by default in the world was, just definitely something that is difficult to ignore. It just creates a lot of challenges because it's a very competitive business, but on the other hand, it creates a lot of opportunities.
Good. So many people in our audience today are in Poland now, and they may be also interested in drug development and or entrepreneurship. You've been successful, more successful than most people in the world, because bringing in not only 1, but 7 drugs into clinical trial for cancer treatments. How do you think you managed that? That's pretty impressive.
Exactly, it's 5 drugs in a clinic.
Okay.
But nevertheless, it's just more than expected from the single PI laboratory. So first of all, you have to look from the perspective, just already not 10 years, but just almost a 30 years career here at the MD Anderson Cancer Center, over 30 right now. So, but you have to be open on the new ideas. And if you have a certain ideas, just you focus on one, but you are not at the same time stop to think about the new things. So you have a focus to develop, let's say, to translate something to the clinics. You have to build some expertise, but at the same time, you just look for some other opportunities. So you never work only on one thing. You work on a few things, and very often later on they interact. Just one helps each other, and so I was lucky to meet also correct people. Just good collaborators. So this is also important things which help to with the certain ideas earlier on. When you have only idea and you don't have a money, just that's always a start. You have to convince, just for everybody here who listen, you have to convince your colleagues first. If they believe in your idea and they are willing to spend some time to do some stuff with you, it means that you are on the right track.
Good. So when you, after you came to MD Anderson, how long did it take you before you got your first compound going?
Compounds going. First we have to start with establishing yourself. You have to have a publication. When I started, just at MD Anderson at that time, just Texas is a oil-rich state. Economy was based on oil primarily, even though the medical center is exceptionally high and as a size number one, the largest in the United States. Nevertheless, just nobody, theoretically, it was patents didn't count to your career. You have to get R01s, you have to get, just publications in order to advance, to secure your tenure. That was just a challenge, and that was the focus practically. So we were initially, you have to go very basic. Fortunately, it's changed with the challenges at the time when the oil price went down completely. Then they have to see, okay, you have a biotechnology and all the new companies start up on the East Coast, West Coast, and we were the third coast, just what they call it here, just on the South Coast. It was, just really not existing very much because we had endowments, just from the oil interest in universities, and everything seems to be working so far quite well. Frankly speaking, we were the first with 3 other colleagues, the first company which we started. It was 3 persons, and these 3 persons were from different countries. There was from Catalonia, just from Spain, a physician. The other one from, just father was from Spain and mother was Jewish, and he was born in Puerto Rico. In Cuba, but just when the communists, they moved to Puerto Rico. So you have that, and then he was a physician also. So we need to have a basic ideas, but the physicians, we started a first company, which was Argus Pharmaceuticals, is not even listed here right now. But in order, that shows you that, just certain similarities maybe to Poland in a certain time. In order to start the company and for us to own the shares in a company, there was a special session of legislature of the state of Texas that state, because nominally we work for the state of Texas. This is a University of Texas. We are a state institution. We have to have a permission to own the stock in a companies which are doing a business with the state, because the company licensed certain intellectual property from MD Anderson, and we own the stock in this company. So that's how behind we were, let's say, in this late 90s, early 90s, very early 90s. So it was like practically it was the 80s. But just later on, once I established my position, having grants and developing certain things which just secure somewhat just position, we looked for the opportunities how to translate the basic science discovery to the fruitful end. So it means to commercialize.
Do you think that the fact that you were in Texas and there was oil and good financing and endowments, did that help your situation?
this really endowment and all these things was not really impacting us directly because it was sort of the function of the university.
Okay.
So it's like Harvard has a big endowment doesn't mean that just people are getting for science just money. You have to it's a hard work to earn some additional funds to bring it, and you are on the top of that. They just pays your duties to the university because they take you have to produce some money revenues because overhead is the revenues for the university.
Right. We'll get a little bit more into funding companies later on in our discussion. So we will talk about drug development. I know you have a couple slides in a few minutes that I'll ask you to put up, but not quite yet, regarding drug development. And you also have been involved with so many things to tie and unite the United States with Poland. And one of the things that you initiated was the 3Cs Center Initiative Program. And we'll talk about that a little bit later as well. Right now, I'd like to ask you, you've been involved with all these drugs that have gone into clinical trials or preclinical. I know you and I the other day talked about what is your favorite drug, and you named your favorite drug after your daughter, Anna. So tell us about that. It's kind of a nice story and tells us a little bit about you and what you've done with Annamycin.
So really that interest in a DNA binding agents stemmed the interest in a certain class of agents, which are the DNA binding agents, anthracyclines. And a best example of that family would be a doxorubicin. And there's several drugs from that family of the class of drugs. It's a daunorubicin, epirubicin. There's just two more which are being used, carminomycin, around the world. Just the common denominator for this class of agents is that they have a serious side effects. However, still. Just it is a frontline drug, doxorubicin, in several, indications. One could be sarcomas, for example, a frontline drug, but just they are leukemias. Just daunorubicin is a frontline drug. You have just multiple combinations for different indication. Despite the fact that doxorubicin and this class, all these agents in the clinics are produce really a serious side effects. Losing hair, alopecia would be probably something the least affected. The psychological maybe problems, but certain indications of the lack of selectivity. But also very serious is a cardiotoxicity. So you have a so-called cumulative dose, just established through the experiments through the years, that just you cannot give more, even if the drug works. You cannot give more or give a repetitive doses because of that potential cardiotoxicity with a serious side effect. They might cure you of cancer, but you will die of the heart failure. Yeah. So that's just was one of the interests to us. Also, there was some development of multi-drug resistance. So it means you treat the person and you have immediate, in a short period of time, develop a resistance, and not only to doxorubicin, but be several unrelated structurally drugs. So that's just led us to that work, which just we have a certain goals in mind, which I project here, to develop a novel non-cardiotoxic agents which will have high activity against multi-drug resistant phenotype. And this is in a clinics in a phase 3 right now, clinical studies, which is quite advanced, and the drug was also in a clinics all over the world in different places, but also in Poland in several sites, and will be tested right now in a phase 3 also in Poland, among others. That's the reason that I brought investors, from the company who licensed this thing from us, introduced them to Poland. We traveled along, and they found that this is just the investment for them also to do the clinical studies, among other places in Poland.
Of the drugs that you've put into clinical trials, how many are in phase 1 versus later stage? That's one question. And along with that, what is your personal opinion about trying a phase 1 trial drug on a cancer patient today?
Let's say, can you repeat the question?
Of course. So the
Phase
first
1?
part is you have a number of drugs in clinical trials. How many right now are in phase 1 versus later stage trials?
Okay, so we have just two drugs in a phase 2 right now, and two in a phase 1 completed.
Okay, good. And just this came up in a conversation the other day in terms of clinical trials that patients with a cancer diagnosis today are looking for, and physicians generally don't want a patient to use a phase 1 clinical trial drug. But that doesn't mean they're not efficacious for that cancer diagnosis. What is your opinion about trying a phase 1 drug on a cancer patient currently?
I would say that it's not the case that physicians don't want to use it. Just this experimental therapies phase 1 very often are not available, just widely. People from, let's say, try to get to this clinical phase 1 studies, because usually to be in a phase 1 clinical studies, not all drugs, but just let's say the drugs which are cytotoxic or really have some certain side effects. And first of all, this is never in humans, so just this is a first studies in humans. So the goal of phase 1 really is not sometimes efficacy, that was a positive effects of such studies, but you are having a people already who practically there is no choice for them. They are told that just you were treated with everything which is right now available for your disease. Surgery, for example, is not the option anymore. The radiotherapy, there's no chemotherapy which would work for you because you already took everything which you could. And if you have the aggressive disease, like just I don't know, pancreatic cancer, or you are late stages of the breast cancer with just let's say certain metastasis to the distant organs, at that moment you qualify for a phase 1 clinical studies. And phase 1 aim is to establish just a toxic side effects, or what they call it, maximum tolerated dose, how much we can give to the patient without causing a serious side effects. During the process, just on the other hand, there is some of the patients might respond, and not necessarily is always a welcome news. And you could see these things in some studies. And therefore, that was one of the reasons that we brought certain, like Annamycin, but some other drugs to Poland, just also because this gives opportunity for these patients to have a chance. Just sometimes you see on a TV that the people are trying to raise the money and just go for some experimental therapies which are not approved as a standard of care to United States or some other places. Some are legit, just real stories. Sometimes people in desperations will go for some therapies which not necessarily are even validated or will not be approved for a phase 1. They go to Mexico or some other places as well, which we should warn the people about such places, because that's a place where you spend the money, but I've never heard about anybody who got helped by such just operations.
Yeah, so you have to keep safety first, I guess.
Yes.
So one of the problems with your career is that you have done so much, and it's so impressive that it's going to be very hard to cover everything we want to talk about in the time that we have today. So we're going to try.
It's a problem.
It's a good problem to have, but it is a caveat to our session today. I have, and I should interrupt to tell our audience, if you have any questions or comments that you'd like to share or to ask Dr. Priebe, please put them in the Q&A, which is at the bottom of your screen. And we will try to get to questions later on in our hour together. I have one question before we move on to more specific drug development information that you can give our audience. You and I have talked about collaboration. You have talked about collaboration, and you've shared the importance of working with other people to move your projects and ideas forward. So can you talk a little about that, and also how for our friends in Poland and perhaps a young faculty or student, how do they initiate these kind of collaborations, especially maybe with the United States because they're in Poland today?
Just one of the things is definitely a meetings. Just obviously, you shouldn't be shy to talk about your ideas and the projects with anybody. Just you meet. Just because you never know who will be the point person later on to tell you, oh, I know this guy who just doing a similar things or something. Just adding. You should talk with that person. So you have to really be quite vocal, and you should have think. I didn't do that fully consciously, just these things, but looking from the perspective, I was able to adopt. Let's say you have to have a prepared longer presentations. Just like say. 2 minutes, 3 minutes, just discuss with key points what you do to some person who has a certain knowledge. I had to learn to talk with a different language to the investors, potential investors who can bring for commercialization. And you should have like I don't know, 30 seconds, 40 seconds, what they call it, elevator talk, a few round sentences to tell about, just a project or something, because sometimes people don't have a much of the time, especially sometimes in the meetings. People, just you go to the audience to talk with somebody who gave the lecture. You cannot talk with him 5 minutes. Just you have to give certain things. Have also your card contacts. Just and follow up after the meeting with such person. Usually, the people who are your seniors, they will be busy. So you have to reintroduce yourself maybe again, or just be patient, polite, but persistent. That would be just a start things. As a good example of the collaboration, I might just tell you that with one of the drugs which we developed, you set up some team of the interested people. I have a neuro-oncologist and I have the pharmacologist, and I bought a lunch, just every Wednesday, and every Wednesday we met in my office, and and we did the project, and just they have their own laboratories, so they chip in with their funds at that time, because we were starting from my idea and what we want to do, and they just bought the idea, and we were able to start the project from ground zero without extra funds. So that just based on idea. And there's as I mentioned, if you convince your colleagues and they put some their own resources and time, that's just a great time. And we've met like that religiously for 1.5 years, just so just almost 2 years. So that shows the determination, the interest, just of your colleagues. Right now, just you might have a easier way to connect, let's say online, which is certain advantage. However, just nothing replaces these personal interactions.
Right. Well, of course, between Texas and Poland, or the United States and Poland, it pretty much has to be online.
In California.
And California. Of course.
It's a few hours flight still from Texas.
Yeah, it's true. We're always open here for collaborations at Stanford and around Silicon Valley. So you have a busy schedule. Do people approach you? It sounds like persistence and a lot of talking with a lot of people would be a good suggestion. Are you open for people to send you an elevator pitch?
I'm getting quite often, just, and
True.
sometimes I say I cannot help, sometimes I can help, just so.
Right.
And but also for example, just right now, just I serve as a mentor for the University of Texas in El Paso, which just there is a funding for the CPRIT part for such, just interactions, that there is a young assistant professor, they need some mentors from the different, more advanced, senior, and state is producing a certain fund, small funds, but it is $100,000 for the 2 years for such collaboration visits and sharing reagents, certain, experiments, just set up and so on, which could be helpful for some startup, careers, especially when the institution doesn't necessarily have that infrastructure yet set up exactly to that level of sophistication as we have at MD Anderson. And we have another one, the same things with the University of Texas, the medical branch in Galveston, which is close by, but is a separate institution, also state institution in their location. And down there, for example, we have an active program in developing antiviral agents right now, just with them. So it's interesting in its own, because on the other hand, they don't have a certain expertise, but we will never at MD Anderson set up, for example, Ebola or COVID, laboratories, which you have to be space-like dressed, just for the whole day, locked in a special facilities which are secured. So if everybody heard about Wuhan, just laboratories, just Wuhan in China, this is, government-sponsored site down there, which all the possible worse bugs, viruses, bacteria are stored down there, and experiments can be conducted and studies down there at that facility.
Well, I want to give you some time now to show some slides and talk about drug development. It's some basic information on the whole progression of drug development, and I think that would be of big interest for people viewing us today.
Okay, let me just start to share these things.
Great.
Okay, can you see it?
Yep, perfect.
Okay. So this is a quite, let's say, long, just process to get from the, let's say, basic science studies, which might lead to certain discovery, so the different steps to getting to that NDA approval, new drug application, just is being submitted at that stage after positive results of the phase 3, in order to get approval from FDA for this to become a approved drug that could be sold freely, just in the United States, but practically at the time later on after the FDA approval, you can get approval in a lot of other countries as a new drug, which would be for the cancer, for the specific indication, could be standard of care. And therefore, that's, just it's not like you advertise too much, because, just after, based on the studies, you have a specific patient, which, for the specific indication is almost obligatory to give this drug. That's how very often, just in this indication, it works. So when you see this from basic science, just definitely as pharmacology, preclinical safety evaluation, all the studies here, you could do some preliminary data, but in order to do IND, investigation new drug application with FDA, just you have to go through the formal studies in this area. And very often here, as you could see, we call it a valley of death. Why? Because you could do something in your laboratory, or your colleagues will do certain studies. Nevertheless, for FDA, it has to be done in a good laboratory practice. It's usually outsourced to some place, and you have to have just here a few $1 million, just to put on a table to do the studies, because your compound has to be synthesized under the good manufacture, GMP conditions, good manufacture practice, which, for example, Annamycin production cost us already like $6 million to $7 million. So which is complicated synthesis. And by the way, right now the synthesis of Annamycin is being done mainly in Italy. Italy is well positioned to a lot of studies in this preclinical or the submission of the or the production of the drug later on for the clinical phase studies, as well after NDA, that they have this manufacturing, which is tightly regulated by the government institution. In Europe, it will be European Medicine Agency.
And
So
why are you calling this Valley of Death an opportunity for Poland?
I wrote this as an opportunity of Poland because, just this is something that can be done. I will start to talk about Italy. It can be done because, just it's, and it could be done in a competitive business because this is, how to say, it's not a discovery, but it will enable and speed up the discovery in Poland. But also it's a good business because in Italy, they thrive on that business of getting, around the world the studies. The pandemic time, just COVID really showed that not everything can be outsourced to China. India is right now just an important site, but the relations with China cooled down for different reasons. So why not to create this business, which could be a business opportunities to produce, let's say, we have excellent chemists, we have excellent, just stuff here. You have to go through certain rules to start to produce a drug under the GMP condition, which will could be not only from Poland, but from all of the Europe, even United States. If there will be such facility, which is not big deal, it's a question of the expenditures, just enough for the money, just we would be doing the synthesis of Annamycin and some other drugs in Poland. Some other drug, for example, we did it with the smaller companies because we need the initial few hundred grams only of the material. We did, for example, in Denmark. There was a company again, just created who fill up all the regulations. So also for the scientists here in a biotechnology, once you have it here locally, it's first of all, it could be less expensive. You can have a direct access to transfer your knowledge down there. Because, for example, the Italians are fine, they do that, but they were not, let's say, they were not smart, let's say, for certain things in a synthesis. We developed a synthesis and all the production was based on our synthesis, which they maybe optimize. And they had a problem to synthesize. They say, no, it doesn't work, it doesn't work, it doesn't work. So I sent the person from my laboratory for 3 weeks, just to Italy and to near Milano, the facility down there. And, everything was worked. Sometimes people have a certain routine. You cannot transfer everything. So it was a key things how they handle, improperly the steps, and they couldn't reproduce the data. They reproduce, and right now this is a large-scale synthesis, quite successful. So here, when you look on a support, I just try to say these things could be done in academia, but a certain element here, you have to get the potential investors. Here in Poland, you might find some support also for that, filling up this gap. Once you get to the phase 1 clinical studies, I put it in blue, then the potential investors and some companies are getting more interested in your project. It's easier to get investors at that stage than pre-clinical. And that's a very good reason. Also here, the other message is that, just people say that, just you have to have the development of a new drug will cost, let's say, a few $1 billion. And then, just you have a decision makers in the institutions, but also in the government. We cannot give anybody a grant of the $1 billion. Nobody ever gets $1 billion for the developing a drug. It comes at the stages. And that's probably, just I might show you in the next slide, this is one here, how the process looks like and how the value of your project will increase. So when you see initially, when you are on that stage of this discovery, practically it is worth nothing. Just you support through the grants, through academia. It's a research. But once you are getting to the preclinical validation, the value starts to increase. Here you have in millions of dollars. So when you get to the phase 1, you already have a value. Something might have as you could see, $30 million. So you can get investors on earlier stages here, just some angel investors or early stage investors, which hope that you will go to that stage, which they will increase the value 5-6 times. That's usual risk for the investor because they spend a lot of money for failure also. So just that gives you the perspective. And here is the chances of your final success. Phase 2, you can have here a value of the company already, as you see, $300 million average. And I can tell you that one of the company, which I was a co-founding scientist, Reata Pharmaceuticals, before they had a very good data on the phase 2, this was not my drug. They licensed one of my drugs, but they discovered by accident that one of the drugs, which was, and this is also very important how the discovery sometimes happen. They develop the drug. They just got in a second round potentially. I was not not excited about anti-cancer properties, but I say, yeah, it's interesting. Let's get it. I was at the committee, just at the time, just evaluating these things. Because we need to have another drug, not only mine, just in the clinics. This drug was not active, but given in phase 1, restore the function of kidney of these patients, which already have a dramatic change. They start to look for the mechanism. It appears a highly promising, just for the totally different indication and totally different mechanism of action that they saw at that time. So you see how the accidental, things sometimes switch and they go for the totally different indication once it's in the humans. And based on the phase 2, they practically got the funding for over $1 billion investment from the big pharma here. So cash upfront and 9% of the company at the time. And they were sold later on for the, just $8 billion, $7-8 billion. So bottom line, that shows you how that, on a different stages, different investors will come and different money will appear. So nobody will give you $1 billion upfront, but just if you progress correct way, there will be new investors and the company will be expanding and acquire, or you create a totally independent company.
So this definitely shows the complexity of this whole process. I will ask you to stop sharing at this point so we can have time to move on a little bit to some other areas. Again, I'm hearing that being persistent is a very important aspect to this whole drug development process, along with collaboration and knowing how to approach funding sources for capital and so on. So what are you currently working on? Like, what cancers are you addressing?
So Annamycin is in a phase 3 clinical studies against acute myeloid leukemia. The other drug, Berubicin, is in a phase 2 advanced studies for the glioblastoma multiforme, which is the most aggressive and deadly form of the brain tumor. And then we did significant efforts in that area. In that area, we have also in a phase 2 another drug for the same indication in a glioblastoma multiforme, which is an inhibitor of the STAT3, which is a signal transducer activator of transcription 3. We have also finished a phase 1 and then going to phase 2 in a pediatric medulloblastoma, for the same indication with the WP1066 compound. We finished phase 1 for the drug which inhibits the glycolysis, and it was done, this is interestingly, in a healthy volunteers in England. That's what it is, because it's a relatively, it's inhibiting a metabolic functions, really a utilization of the sugar, which drives certain, just tumors, but also, believe it or not, is a critical metabolism of glucose and mannose for viral replication. So viruses to replicate, just they require the higher energy. Once they enter into the whole cell, they change the metabolism, which is highly dependent, just on a high efflux of the glucose and production of the mannose also, because they have to build the glycoproteins, but also just all the building blocks, very often come from the glucose. So when you cut this down, then the virus, you stop their application, or if you give them a false mannose, they will have a misfolding of the proteins. They cannot, just infect. The infectivity will change. So this was a drug which was supposed to go for a COVID, and at that time, and so that was an interesting concept in its own right. COVID ended, but there is right now progress, exploring these things for the cancer.
How have you managed all these years in your career so far to juggle what you've done in academia, what you've done for entrepreneurship and starting companies, drug development? How have you managed to do all that and bridging the gap between academia and the business side?
Just you cannot get involved in a business, just if it would be one company and one, just my laboratory, maybe it's possible. With such, just multiple companies and then multiple products, you have to really, you create, there's a team, the companies, just has its own responsibility, especially that they are public companies. So, just usually I'm staying for a certain period of time as a chairman of the scientific advisory board. I'm involved in a daily, very often discussions, because, for example, when we do a clinical studies, very often questions will come, or certain things are unexplained, or certain challenges will come from the clinics to refine certain things, formulations, for example, later on. Just even if we outsource certain things, you have to be on the top of that, because outsourcing looks like, you give them, but this is a service. If you don't supervise with your knowledge, they screw this up, because they say, okay, we did the studies with the standard of care, you pay $500,000 and doesn't work. Why doesn't work? They are not interested, or what mistakes they made, or how they set up the experiments were that they didn't get the correct answers. So you really have to have within a company also certain expertise. And this is like just a conference calls. Thanks God right now, just that we have also online meetings. If you have a project, you have from the different outsourcing and different challenges down there, the productions, always something comes up. For example, try to imagine that we are doing in Scotland, never again down there at that place at least, we did a formulation of the drug, Annamycin, and they were slow with certain things. So therefore, instead of finishing with a certain period of time, they left these things overnight to finish the next day. So they finished these things, but it was already decomposed, because they shouldn't stay this way.
That's frustrating.
So bottom line, the whole batch, which was $600,000, this batch, just went to the drain.
Down the drain.
And we already prepaid, because, just usually that's the things, practically lost case. And so sometimes you have to be very careful. You go to these places, you checked, just the sites. You have to be very careful who you are dealing with. So a good example, and other things, company, this is independent decision, went to Australia, because there are some nice credits for the company because in tax and so on. Sounds good, but Australians, the company was outsourcing certain things in China, which they didn't tell you. And then, just they went bankrupt during this time. $500,000 was lost. So just you have to be very careful just who you are dealing with. So just it requires, a lot of things. Fortunately, here I'm doing these things from the second line, but sometimes you have to go for a moment for the first line, like where is the problem? But, but also we do a lot of exploration and looking for the new indications as well for the Annamycin. We develop, for example, right now, we are going after acute myeloid leukemia, ostra białaczka szpikowa po polsku. So we, just look for the other indications, highly promising other indications for Annamycin as well.
I don't see questions yet in the Q&A, so we have a few more minutes. I'm going to ask you one more question, and then we're going to move on to sort of our last topic, which is the 3 Cs initiative. We talked a little bit about academic training. What do you recommend to train fellows in Poland, and so they have the necessary tools to do what you've done at MD Anderson, and what can be done globally?
Just definitely you have to look for, just a interesting place around the world, which you could gain more experience. That's one thing. Learn new things, and so postdoctoral fellowship is a critical, and it doesn't need to be at one site. It could be in a 2 sites. You can have 1 year here, 2 years another place. Don't be afraid. This is the key moment that you also develop certain relationship with certain people, just who are experts in a certain fields, and they could be, just you're also later on a mentors. Very often, just there was the idea which I was trying to propose to the Polish government that they will create some fund, because, for example, you have people from France, from Germany, coming to MD Anderson, from Japan, from Korea, from Egypt even, just coming with their own government money. Therefore, you can apply to any place you want, just for the fellowship, just and to learn, and very good institutions. So that's the key. And I would say that for such person, younger person, if they want to return back to Poland, they should have a special pool of grants without too much of a trouble. You apply together with your mentor. So certain things you might conduct later on in Poland, and that is beneficial because certain things will happen faster, and it gives you additional validation of what you do and certain verification, and the continuation of the mentorship after this 2 years of the postdoctoral fellow. You still need for the startup some energy, and money and continuation.
That sounds like a good recommendation to give a young investigator today. You've done an awful lot in terms of creating partnerships between Poland and the United States, and particularly MD Anderson and Poland. I know you showed the president of Poland and the president of MD Anderson coming together. These are big things to have put together. It's remarkable. But one of the things that you did was to start this 3 Cs Cancer Initiative program, and I'd like you to talk a little bit about that. We don't have a whole lot of time left, so we'll have to watch a bit in terms of the time left.
Okay, it looks like just we are on a short, there's 12 minutes left. Yeah. So let's say that we started first with creating a partnership with the institutions or the scientists to start to interact. And this was a program which we set up the sister institutions with MD Anderson to increase the interactions between scientists, share the experience. The motto of MD Anderson is to making a cancer history. So it means that we are fighting, just to improve the treatment in state of Texas, in United States, and all over the world. So this is one of our mission, is to share our expertise and share our knowledge how to improve the treatment of cancer. So within that, it was possible to create such, just interaction. Here was the President Duda, just of Poland, President of Poland at that time. He showed some interest in oncology, and it started with the invitation of the leaders of MD Anderson to Poland and visiting a different cancer centers. And the president in his palace organized a meeting with the leaders of the cancer institution. So it was a nice meeting with everybody, leaders. And then, let's say, vice president of MD Anderson for clinical studies, research, the vice president for international relationship, some leading clinicians. We travel from place to place in Poland, personally meeting, giving lectures, and that was the startup. And later on, it wound up in a exchange, collaboration, and creation a new plan, government plan, just with the Minister of Health, just of Poland at that time, initiated by president, of creating the new plan to treatment the patients, oncological patients. It was a big improvement, a big investment during these last few years, which expanded the center in Warsaw, just expand dramatically and several other institutions. They build the proper facilities, updated facilities to treat the patients. And there was an interesting collaboration that people start to know each other. Also, for example, in a prevention, we have two persons which were involved in a prevention, especially Jeff Gershenwald, he's a melanoma specialist, and we introduced several regulations, state of Texas, different states. United States, state by state, is doing its own regulation, not centrally, which regulates, let's say, tanning salons, because there was studies from MD Anderson and some others, just demonstrated that there is a significant increased risk, just when you attend to tanning salons, and very often the kids were brought by their parents to the tanning salons. So we reintroduced 18 years old, you have to be conscious decision, regulation, just these machines, just what was the exposure, just to the patients and so on, or to the person who would like to be, attend the tanning salon sessions. So this is especially regulated. So here, President of Poland took legislative initiative, and we have this regulation based on the United States introduced in Poland. This was mentioned in a yearly report of the World Health Organization, even, that you have the government in introducing regulations which are working for the benefits of the patients, cancer prevention regulation. So it is a lot of small things, but what was important also, there was some collaborations which were initiated at the time.
Yeah, that's fantastic. I'm going to break for a second because we do have a question in the Q&A box from Christoph. In our group at the Jagiellonian University, we have obtained a photosensitive analog of cisplatin, which reversibly changes its cytotoxicity under near UV and visible green light. What is your opinion on applying such a photopharmaceutical approach, that is to say, changing the activity of drugs with light in oncology?
Definitely this should be explored further, but just in more details, and I will be more than happy to look at that. For example, I was 4 days in Krakow in November, and I usually, I'm 4 or 5 times a year in Poland, and usually in Krakow, I'm also, just for a few days, because, I have in Warsaw and in Krakow some sites that I can, just relax and stay for a moment. And we have certain collaboration with the University, Jagiellonian University, with Professor Romanowska-Dixon, and certain things we test together for the uveal melanoma, for example. But this is an interesting concept it depends, how stable that drug is. If it's, have its own, just metabolism rapidly and cytotoxicity without radiation, or this creates a stable, prodrug which only is activated, just upon radiation. So, and it should be checked in a certain models, and certain type of the tumors might be very useful. Definitely.
Christoph has put another comment in the box. We have patented the compound and described in recent issue of Journal of Medicinal Chemistry. He puts the link. I will type, let's see, an answer to say, here is my email address.
We can share my email. I think it was shown or not, or I can show, my email shortly here. So just so.
If you'd like to type it into the Q&A, Dr. Privé, that would be great. If not, I just typed my email address, and Christoph, you can send either of us or both of us further information and the link to your article.
I just send my.
Okay, thank you.
My.
My God,
email.
thank you very much, he said. Great.
And,
right.
So I'm open, just with pleasure, just to look for some novel things. And especially that, just, okay, if there is a certain platform technology, you did it for platinum, maybe this will be applicable to some other drugs. We can just bounce some ideas, just it's a proof of principle for one type of the drug, might be applied maybe for some others, just we can discuss some, just ideas. Definitely. I'm quite open.
Well, that's very nice of you. Thank you. And I'm sure that he will follow up. So we only have about 4 minutes left, which again, the time has just gone so quickly.
If somebody is, let's say, unwilling to type and would like to ask the questions, probably, I don't know if it's possible, just you are welcome to ask the question.
Thank you, Dr. Privé. Recently, you were just honored with the Lifetime Leadership Award through the Polish Chamber Texas committee, and tell us about that. That's quite an honor.
Just I would say that just I didn't look, but just it was a nice, pleasant recognition of certain activities which you mentioned, and but it's. I appreciated the recognition, obviously, but it comes and goes, and just doesn't change my lifestyle. You know what I mean? So just we still work the same, do the same things every day. So just so it was nice that they recognized, and also just in Poland, honoris causa title, just was also, just in November last year, just we have a nice event. So that's interesting, just recognition, including, as you mentioned, just I'm a full professor, which is a professor, which in Poland they say Belwederski, because it's usually given by the president of Poland, nomination, just so I got it from the President Kwaśniewski. I got some recognition, some medal, Odrodzenia Polski, from President Komorowski, and I'm in a Narodowa Rada Rozwoju with President Duda. So you have to, you try to work through the years for the best, just relationship between Poland and the United States. But also I have to mention that I'm a trustee of the Kościuszko Foundation through the many years, and therefore, just that's creates a certain opportunities, just for the creating some exchanges between the institutions in Poland and United States. There is some grants which are given every year by the Kościuszko Foundation. So just probably you should Google as well. And usually there's some applications, and the Kościuszko Foundation is selecting the best candidates for that fellowships, just in United States. And also right now there is a branch in Poland, in Warsaw, of the independent, let's say, part of the Kościuszko Foundation in Poland. And I'm also trustee at that Polish foundation. So that's where to look.
Well, you're certainly being very modest about all your achievements over these years, and I'm also sure that you would love to have some young scientists in Poland collaborate with you to continue and help out with everything that you've been doing. I wonder if you have time to sleep at night. So
It's sometimes, just you stay quite long.
yes, I'm sure. This has been such a pleasure to have you here today and have this conversation together, Dr. Priebe. Do you have any final, in our last sort of 30 seconds, do you have any final comment you'd like to make?
Just I would say, don't be afraid of new ideas, and whatever you read and seems to be that a little bit, statements which might not be correct, but the people say that it's, let's say, white and you think it's black, verify these things, just because few times we found a very good idea, but being suspicious that, just somebody makes on a one experiment a very general statement, and very often is, just adopted as a universal truth. And that could be smaller things, bigger things, but, might lead you to the novel discoveries and novel things.
Well, Dr. Priebe, thank you again so much for being here today. It's been a pleasure, and I'd like to thank everybody from the Poland and Silicon Valley Center for attending our webinar today, and I hope we'll have some opportunity to continue our conversation in the future. So best for the rest of the day, and thank you for attending.
Thank you.